Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture number 44, that is Metabolic Engineering of Carotenoid Pathway, and here we will discuss the golden rice story. So, it is about the rice. So, engineering provitamin A pathway in pathway rich engineering provitamin A pathway in endosperm, I will correct it, and improving the nutritional value of golden rice through increased provitamin A content. So, it is all about the rice and why golden rice. So, why rice? Rice basically a staple food of Southeast Asia. For and particularly in Southeast Asia, the people who are below the poverty line, they mostly consume rice as the major diet. So, actually they are not getting other balanced diet. So, the Southeast Asian population those who are below poverty line, they mostly consume rice as their main meal without a balanced diet. Proper balance diet, and as a result what happened, continuous consumption of rice in 2 times or 3 times a day, eventually that led to the formation of diseases like night blindness, which is called xerophthalmia. And the reason for night blindness is basically the deficiency of vitamin A. That means, the Southeast Asian population are mostly suffering, a majority of the population or sizable population are mostly suffering from night blindness because of their food consumption pattern, which is only rice, because they did not have wealth to get a balanced diet, and as a result of that, with time what they develop vitamin A deficiency, and that is why they are under under the attack of this xerophthalmia disease. So, so now to solve this problem, if the deficiency of vitamin A can be solved by adding the pathway of vitamin A in the rice plant, or in other word, is it possible to create nutritionally rich rice containing provitamin A. Now, I need to tell you briefly about this pathway of provitamin A. Although I have discussed the carotene pathway, but this part I need to re-emphasize once again. So, beta lycopene, we have discussed lycopene. So, lycopene, from here it makes beta carotene. So, this beta carotene if we consume food containing beta carotene. So, in our body system there are enzymes which convert beta carotene to transretinal and that subsequently converted into transretinol. And then it will convert it into trans-retinoic acid. So, this pathway operating inside the human body. So, that means human body has the enzyme to convert beta carotene into vitamin A. So, for which one is vitamin A then? Vitamin A is this one, transretinol is called vitamin A, and that is why beta carotene is called pro vitamin A. Okay. So, this is brief about the concept. So, eventually what needs to be done is basically the pathway to be incorporated, so that the rice plant will produce rich antioxidants like lycopene and beta carotene. Okay. And then what will happen, that if that rice is consumed, then our body system will convert the beta carotene into transretinal and transretinoic acid. So, and then that will be the vitamin A pathway, and then vitamin A deficiency can be fulfilled by this diet supplementation. So, keeping this in mind, two scientists they collaborated at international level. So, first of all we need to know that whether it is possible to transfer the pathway in rice or not, that is the first question. First of all, that whether the pathway exists in the rice or not. No, rice, particularly rice endosperm, because that is the edible portion. So, question is whether in the rice endosperm the carotene pathway exists. Answer is no. Then next question is, what about the substrate? Whether GGPP is available? In the endosperm? The answer is yes, but it is for different purpose. So, GGPP is not taking part into the carotenoid pathway. That means, the pathway is basically non-existent. So, you have to basically transfer the whole pathway. Just to remind you that from GGPP it makes phytoin. I am only showing the main compound, then it makes zeta carotene, then it makes lycopene, and then in the next step it makes beta carotene. Obviously, transretinol cannot be made by the plant, that I made it clear. So, this is the pathway. So, only thing is that GGPP is available in the rice endosperm, but not this. So, then what you need to do? You need to hire the genes. So, you need to hire a PSY gene. Okay, you need, maybe you need to hire PDS gene. You may need to hire the ZDS gene. You may also need to hire the LCY gene. Okay. Then only whole pathway can be operated. Now, putting all these genes together technically, whether it is possible or not, that is again a question. So, there are first of all, whether these genes have already been characterized in other system or not, that is important. So, and for your information, a scientist, a professor at the University of Freiburg so that is in Germany. His name is Peter Beer, Professor Peter Beer. His group was working on this carotenoid biosynthetic pathway. So, Peter Beer basically characterized the genes like PSY from daffodil. The scientific name of daffodil is Narcissus pseudonarcissus. PSY is from daffodil, and also they have LCY gene also from this daffodil. And they isolated the gene from CR from a bacteria called Erwinia uredovora. So, this is I have shown in one of the previous classes that putting this gene is basically making phytoin to lycopene in one step, that is CRTI. So, this team collaborated with another group at Swiss Federal Institute of Technology. I write the full name, Swiss Federal Institute of Technology, ETH, yes, I remember, ETH Zurich, that is the German name, ETH Zurich. So, there Professor Ingo Potraikas has developed a technology. What is that technology? It is basically the technology for rice transformation using Agrobacterium tumefaciens. So, this is very important because why? Rice basically a monocot. All of us know rice is basically a monocot, and monocot are usually recalcitrant to Agrobacterium tumefaciens infection. So, dicots are easily transformed by Agrobacterium, whereas monocots difficult, but Ingopotrichus developed technology where rice gene can be transformed, and not only that, he has also have the technology for putting multiple genes in rice plant. So, therefore, these two scientists, they basically made a proper collaboration, and that collaboration eventually, so the collaboration between these two scientists So, why collaboration is needed? Because Peter Beer will supply the genes which to be put, and Ingopotrichus will have the technology for transferring the genes in the rice plant. Okay, now, let us see that what has been achieved with this. So, let us go to the next board. So, that is rice transformation. So, they have used two different constructs. One is I will rather say one is the construct for single transformation. So, single transformation, and another is the co-transformation. So, they have used different constructs. For single transformation, they have used a single construct, where they have these genes. For example, what they have decided? They have decided to take the PSY and they have decided to take the CRTI. Okay, and obviously, when CRTI is to be taken, here transit peptide to be added, and then these genes are putting in a proper vector. So, that means that contains promoter and terminator, and it also contains the antibiotic resistance marker gene. So, all these things I am not discussing here. So, along with this you have a gene for kanamycin resistance. So, what I can say NPT2, which gives kanamycin resistance to select the transformed plants from the untransformed one. So, see here these constructs. So, they have put the PSY and CRTI. So, what is expected? That it is expected that if you use this single transformation construct, then the if the genes are properly expressed, then GGPP which is available will be converted via phytoin to lycopene, and lycopene should accumulate in the rice endosperm. Okay, and let us see the co-transformation construct. The co-transformation construct, they have used two construct. One is the phytoin synthase, and the bacterial isomerase. Okay, see it right, and obviously the promoter is there anyway. So, the space is less, so I have to delete this, and terminator, you have a promoter here, and then you have the gene for phytoene synthase. This is construct 1, and in the second construct what they have put? They put a LCY, that is LCY. I put it here red. No, I should not, rather I put it in the green only. Along with the antibiotic resistance marker gene. So, here also both promoter terminators are there, and so this construct was named as PB19 HPC. And this one, the top one was named at PZPSC, and the bottom one is P stands for small p stands for plasmid, ZLCYH. So, here basically two constructs in two plasmids and two plasmids in two agrobacterium. Okay. Here single construct in one plasmid in agrobacterium. So, here what they have done basically, they have put these two constructs into agrobacterium, they grow them together and then they mixed it up and then they inoculate. So, the for plant transformation work, they have used as a immature embryo as the explant. So, this explant was basically infected with agrobacterium, two different cases, right, separately. So, this is event number 1 and this is these two are event number 2, and then after that they put it in a proper medium containing antibiotic resistance, and then from there they have screened the putative transform plants which contains the desired gene of interest, and then through further selection they finally selected a few populations of both the types and then they analyzed. Now, the question is this, what they have found. So, in the second case, whereas the case number 2 what is expected, that In case number 2, what is expected, that GGPP will make phytoin, phytoin will make lycopene, lycopene will make beta carotene. Okay, I just beta carotene. Okay. That is basically for the job 2, and this is for the job 1. I think it is clear. Okay. Now, what they have achieved, that is the question. So, what they have achieved is basically in the first case where PSY and CRTI gene was expressed, where it is expected that they should get the endosperm which should be red in color, because the endosperm should accumulate the lycopene. But what happened, that they did not find any lycopene. Instead, they found that in case number 1, no lycopene detected. Instead, beta carotene was detected. And along with other carotenes. So, why it happened? That is very difficult to interpret. So, in the first case, no lycopene beta cyclase gene was inserted. So, therefore, lycopene should form as a result of CRTI, and it will not get any opportunity to convert to beta carotene. But what happens, no lycopene was found, but beta carotene along with some other carotenes were detected. So, the question is that whether the beta cyclase gene was there in the rice which got induced, it is still not clear, but it is something very interesting fact when they have analyzed. However, in case of 2, that is case number 2, there what they have found, they have also there they have found, of course, no lycopene, but beta carotene as the dominating pigment along with other pigments like zeaxanthin and so on. Okay. So, that means in both the cases beta carotene accumulates, that is the good thing. In case of 2, approach 2, where co-transformation was used. So, there it is expected that it should make beta carotene, okay, provided the genes from both the plasmids are simultaneously transferred and integrated in the proper site. So, that the transgenes will be transcribed properly and make the functional protein, and eventually it get the GGP from the endosperm, and then it makes the product. So, which has happened in the first case, it was really astonishing, but there somehow some induction of enzyme which converts lycopene to beta carotene as other. So, in the second case, beta carotene was dominating along with zeaxanthin and some other carotene pigments, and okay. And from the endosperm, the carotene level, this I must say that 1.6 microgram per gram of carotene was achieved per gram dry or we can say beta carotene level. So, this is a pretty good amount, but although not achieving the daily recommended dose, but the outcome was really interesting and outstanding, and that is why this paper published in the famous journal Science. So, I will give you the reference just a moment. The paper published in the famous journal Science in the year 2000, volume number 287 and page number 303 to 305. It is a short paper, but details are described in the supplementary. First author is E et al., probably a postdoctoral fellow who worked it, but here both the Peter Beer and Ingo Potraikas were the corresponding authors. So, here the title of the paper was Engineering the Provitamin A bracket beta carotene biosynthetic pathway into carotenoid free rice endosperm. Very clear title. So, rice endosperm was carotenoid free, only substrate was available, the pathway was transferred and it produces beta carotene. So, this is an outstanding achievement, and this was done by a collaboration between the ETH Zurich and University of Freiburg, and this is called the golden rice 1, and after that I just take few more minutes to finish this class. I just give you a brief overview that once this has been achieved. So, this golden rice what they have developed, they have not patented it. So, they made this technology available freely anywhere in the world, and lot of trials were made in developing countries, but this basically boosted the private players, the industry players to join in the group. So, they have more resources, more money. So, they started doing venture, and as a result of that they have achieved golden rice. So, I call them golden rice 2, and in one such construct they have achieved the carotene level up to 6 microgram per gram dry weight, whereas what I have shown with the other one is 1.2. So, this was achieved by using this. I just put the construct briefly. They put PSY, and along with that they use CRTI, and they have used synthetic transit peptide, and the promoter was different. So, I will not go to that details, whereas in case of public laboratory, the promoter whatever used for CRTI was 35S promoter, but here they have used endosperm specific promoter. That is only the difference. They have used the endosperm specific promoter. So, this was glutenin B promoter, and the phytoin synthase was also used under the control of a endosperm specific promoter. Promoter. So, this both are basically endosperm specific promoter was used. and here this CRTI basically they have used the synthetic transit peptide fused with CRTI. So, this was the synthetic TP with CRTI. Okay, so this is this construct they have named as POGR 011, and this they have achieved by co-transformation, and because co-transformation means the antibiotic resistance marker gene they put it entirely another plasmid, so that in the subsequent generation it can be segregated out and you can get the transgenic plant containing only these two genes. So, that is why co-transformation was used, whereas in the other plasmid only the antibiotic resistance marker was used. And finally, before ending what I will tell that another construct was made by the Syngenta group, and which leads to the achievement of 37 microgram per gram of carotene. So, from 1.6, 1.2 to 37. So, this was achieved by again using the construct. So, I just put it here. One is they have used the phytoin synthase, and this phytoin synthase was Narcissus, pseudo Narcissus, but this phytoin synthase is basically from maize. So, it is basically Gia maize phytoin synthase. So, that is a monocot source, and otherwise the promoters and terminators were kept same. So, that is one. Along with that, so that means the promoter was endosperm specific promoter, and along with that what they have used? They have used the normal CRTI with the normal transit peptide and with the normal endosperm specific promoter. And of course, the one here the antibiotic resistance marker gene was there. So, here one more thing I would like to tell just before ending. So, they used phosphomannose isomerase as a positive selection marker, whereas antibiotics are basically marker genes are negative selection marker. So, mannose was used as the medium, so that phosphomannose isomerase, if the gene is there, it can degrade the mannose and make the and convert it into fructose, and which is available for the plant to grow. So, this construct was they have named as PSYN 12424, which is GR2. Eventually, this is called GR2. So, this one, the first construct, this is called GR1. So, okay. So, I have made the golden rice 2 because use the term at the beginning, because the aim ultimately they have achieved GR2. So, they have achieved GR2 from GR1. So, GR1 led to the accumulation of 6 microgram per gram carotene per dry weight of endosperm, whereas here it accumulates up to 37 microgram per gram. So, that is a very outstanding achievement, and this was achieved by the private laboratories, Syngenta laboratories. Okay. So, this is all about the golden rice story. So, these high carotene content golden rice are basically exploited by the companies for profit, and those who have money they can buy, those who do not have money they have to go for the golden rice which was created by the public laboratories. So, with this I just show the last slide, which is the picture. So, as you see here, this is the wild type rice, that is untransformed. This is the golden rice 1 and this is the golden rice 2, and here it is showing again in another way. So, there is indeed distinct difference between golden rice 1 and golden rice 2. With this I end this class, and this is a very nice example of metabolic engineering, and I hope you have enjoyed. Thank you.